Okay, we are mainly working on live cell distinction, and we use the fluorescent molecules as a tool. So, the Vivian introduced, my background was organic chemistry, maybe a little medicinal the flavor. But since I moved to the postdoc at the lab in Peter Schultz's the group, I started some kind of a fluorescence-based imaging as the part of my research work. And later, I thought if you can identify cell-selective probes, then it could be really nice. Our body is mainly the composed of cells, and majority of the cells in our body, they have different shapes, but approximately they have about 10 micrometer size. Maybe the largest number of cells in our body is a red blood cell without nucleus, and biggest one is an egg cell. It's maybe 10 times bigger than regular cells. Longest one may be the neuron cells with an axon length of maybe up to 1 meter size. Now, how many different types of cells we have is not very clear. So whenever I encounter any biological problem, my first reference is Wikipedia. And if you go to the Wikipedia and type the human cell type, they list up to about 200 cell types there. So that means up to 200 cell types, at least we know a little about them, and even we could give a name to them. Okay? But that does not mean that we have only 200 cell type. We may have a minimum more than 1,000 recognizable or distinguishable other cell types. Now, if you look at the cells under the optical setup, the bright field images due to the diffraction limit, even it is difficult to see the small organelles inside the cell. And with a little better the trick like a phase contrast or maybe dark field, we can see a little better. But still, even looking at those, the mitochondria or nucleus or the ER structure is also quite challenging. Then how can you distinguish two different cell types? That was the question that we asked. So let's say if there are T cells and B cells, there are lymphocytes. Both of them are originated from same ancestor, and they look similar in shape and size. So the common way to recognize the difference between these two cells may be surface markers. There are some different proteins expressed on their cell surface. And if you use proteomic approach or maybe genomic approach, and if you can figure out specific protein on one cell type, not the other, then that type of the target we call biomarkers. So biomarker is the partner inside the cell, and probe we define which can recognize the biomarker and report to us. And there are also another term, sensor. Sensor is a changing its a color or intensity kind of things depending on the environmental changes. So in a sense, sensor is kind of a small part of a probe. Probe, even though it doesn't show any changes, it still can report. Then we can call it as a probe. So some people is kind of sometimes confused with these two terms, biomarker as a probe, but we define biomarker is a biological the signature inside the body. Probe is something we put in from outside. If you have a biomarker, now we can call or we can start so-called hypothesis-driven approach. Here the hypothesis means most likely it's a biomarker. We know the biomarker, then maybe one trick, one way is we can use biological library and then make antibody and then label that antibody with the fluorescence. Then this antibody we can call maybe the B-cell selective probe. So probe sometimes, in case of the antibody, it can be even bigger than the epitope size of the biomarker. So it does not mean that always the biomarker is bigger than probe. Sometimes it can be opposite. So we define those terms by functional roles. Now, if you have that kind of a biomarker on cell surface, we call those biomarker as a CD, so cluster of designations. And if you have the antibody for that, then we can use that after the labeling with the antibody. Now, how many CDs we have so far? Interestingly, more than half a century of the time, thousands of the immunologists, they tried to select the good cities. And so far, the most updated data shows that only 371 cities are approved or established. So that means if you have more than thousands of different cell types in the body, using single antibody defining one cell type is basically impossible because we don't have the high enough numbers of the tools, even though that is the most sophisticated tools we have. That does not mean that we have the more other biomarker. Sometimes the biomarkers are inside the cell, and even though in this case we have an antibody, but we cannot use the antibody for live cells because antibody cannot penetrate the cell membrane. In this case, maybe one way is we fix. Fix means utilizing cross-linking chemical and then making those cross-linking between mainly the proteins. Still, the membrane is there, so now you need to add the detergent and dissolve some part of barriers, and then the antibody can go inside the cells. This technique has been utilized for, let's just say, for the fluorescence, the imaging, and if you use antibodies, by the way, most likely we want you to get as many as of different colors of staining, but still, nucleus imaging is most likely is a blue color. And this green color is antibody for tubulin. And we can see one is the healthy muscle cells with a connected tubulin structure. The other is a fragmenting with a tubulin the attacking the drug treatment. And also our eyes can see at least the multiple colors, RGB. So we can use the multiple antibody to show multiple colors. And if we can overcome the optical transparency issues by clearing technique, even by using 3-4 antibodies, even you can look at through the connection of neurons in the brain. Looks like it's really dynamic and something is moving around, but actually the situation is we fix the cell. And this is kind of a ghost image and eventually remove the majority of those components, just leaving out those cross-linked protein structures we see. So here we thought maybe as a chemist, we can do something better for live cells, especially when the target is inside the cell. Now, if you look at those live cells cell membrane, now the composition of live cell the membrane mainly made by the phospholipid, as we know. And phospholipids, if you look at those head groups, majority of them are negatively charged, or at best it's a neutral. We don't have positively charged the phospholipid. Still recall, outside is a slightly positive, and inside is a slightly negative. That means maybe inside is further negative comparing to outside. It's relatively outside is more positive, but it does not mean that the surface of a membrane is really positive. That's why if you have any kind of a negatively charged dyes, due to this the electric repulsion, the dye has a very low chance to penetrate through to go into the cell. In contrast, positive charged dyes, at least they can be attracted on the surface and they have a better chance than negative charged compound. Neutral, yeah, they are neutral. So these compounds, depending on their physical properties, so-called drug-like property, Lipinski rules, if they have a reasonable range of hydrophobicity, they have a chance to get into the cells. And then those compounds we can select or modify, which can recognize a different part of the body, different part of the cell, let's just say mitochondria, or maybe using those hydrophobicity, ER, Golgi, or maybe DNA binding affinity to make it into nucleus kind of things. Now, based on this, we thought maybe if we make diversity-oriented library, the idea was like this. Any organic molecules, any organic dyes, they need to have ability to absorb the light. And to do that, either we can use metal complex or maybe conjugated systems. Now, these dyes, if you design them with a lot of the dynamic motions, they may lose their energy by vibration or other irradiation of heat. But if they bind to the target biomarker, then maybe that binding event can suppress those motions. Then this molecule potentially can be turned on. That was our original idea. So to realize that idea, let's just say this green molecule is fluorescent, the fluorescent, and this is rhodamine. These molecules, they have a carboxylate group here. That's why they block this rotation here. That's why most of those molecules has a bright fluorescent molecules. Starting from this, we intentionally remove this carboxylate, then this phenyl group starts to rotate. Seems like this is a minor event, but the quantum yield of almost 100% now drop here down to maybe 1% or 5%. That effect is huge. So utilizing this kind of concept, we tried to make as many as possible the fluorogenic molecules utilizing any scaffold known so far. So in Singapore, using quite the generous support, we tried to collect many people, tried to make many molecules. I said many proudly. Well, we made a little more than 10,000 compounds. It's not mixed, they are not mixtures. They are one by one purified compound. And that means if you keep this molecule into 96 well, first and last column is empty, then this is one plate is 80 compounds. Then 10,000 compounds is about 150 plates of this. So you can imagine how much efforts we put to build this kind of libraries. Using these toolboxes, we try to attack to discriminate the different cell types. So these two cell types, let's just say A cell and B cell, especially if there is no biomarker, then you cannot do hypothesis-driven approach. Well, if there is no biomarker, or I'm trained as an organic chemist, it's only I do not know about the biomarker, but other people may know. Still, it doesn't matter. So in this case, we can approach with any cell type in hands. Then we can still try the screening-based approach. So this approach we call discovery-driven approach. Sounds fancy, but actually we don't know much about that. So let's just screen first kind of approach. So to accelerate those screening, important thing is how can we manage those compound in an efficient way. If not, testing 10,000 compounds easily you can scare your student or your the working people. So we tried to make it as simple as possible. So if you have 80 other probes in a plate, we tried to make that copy exactly same the format into 384 well format. So that means one compound can be designated into 4 wells, right? Here, if you use a multipipette by zigzag the transferring, you can transfer these molecules manually without a machine. And then one compound can be into 4, then you can grow A cells and B cells side by side to remove, to minimize the batch-to-batch variation. And then still you have room to do duplicate experiment. So with this kind of a format, now you hope that one compound stains only one type of cells, not the other. So this is our the primary the screening format. And utilizing this over last 15 years, we tried to analyze or select different type of the cell selective probes. So before I move back to my home country, we found about 20 different cell-specific probes against many different type of cells. Around the time when I decided to come back, we started to think about the target for tumor microenvironment. Tumor microenvironment, it has multiple other players. Let's say if tumors start from tumor stem cell, there is a very small population, but by changing their fate into differentiated forms, and they started to build up those bulk by breaking out our immune defense system. And once they make this bulk of the structure, now inside of this the tumor, they have a problem for the supply of blood and nutrients, and they recruit blood vessels inside. But immune cells did not give up yet. So macrophages or maybe other type of immune cells try to suppress those tumor activity, and also some of those other dendritic cells for regulating tumor function, NK cells or T cells and B cells. They are trying to manage these situations. Also, in tumor tissues, not only those the immune cells and tumor cells, there are changed the fibroblasts, the so-called cancer-associated fibroblast. Multiple cells are connected together here, and also their functions are kind of changed because they do not attack tumor too aggressively. It's kind of managing those situations. This is a special environment we call tumor microenvironment. So there are multiple targets here. The main players are either tumor cells or immune cells. So we started to look at to get one cell selective out of this multiple the complex and try to get to those selective probes. So I prepared four small the examples for today's presentation with all different mechanisms. So first case is a tumor initiating cells, so-called cancer stem cell. So following the platform, as I told you, using these other compounds, and then tried to test them in differentiated tumor and cancer stem cells. And then we found one compound and gave the name as a Ty tumor initiating cell yellow. At least you can guess maybe the compound color is yellow here. And again, the nucleus is blue. So left side is this is a differentiated tumors, and right side is a cancer stem cells. So it seems like it's quite selectively staining only cancer stem cells. And if you grow those tumor sphere, looks like they are pure, but actually some cells are stained by the Ty, but some cells are not. Now, CD166, the antibody originally known to be selective to the tumor stem cells, but if you can see easily, some cells, they are not Ty positive, but they are still CD166 negative. So we suspect maybe this means Ty positive negative, CD166 positive negative. Seems like a Ty is more selective than antibodies. So small molecules, even though we are claiming that we wanted to build antibody like the two boxes, but antibodies is selected from millions minimum. Our compounds maybe from 10,000, maybe as a synthetic compounds is quite big, maybe biggest for now, but we cannot usually compete with the antibody for the selectivity, but sometimes it potentially work better. Once you have this kind of the tumor stem cell, the compound, then you need to prove it. So we tried to select those cancer cells by Ty. So Ty positive cell, which is presumably the cancer stem cell. Ty negative is the other cells. And then if you inject this into other animal, then it clearly shows that Ty positive cells make up those tumors much bigger than controls or the negative cells. We tried to do the limiting dilution experiment with this. So started with 1,000 cells, and positive is bigger, negative is smaller, but still there is some tumor is forming. We diluted further and further and diluted down to 1 cells. So this is dilution experiment. So even though we counted as 1 cell, but it's possible it may have 2 cells or no cells, right? So in this case, we tried 8 animals, half of them with a single cell injection, they can form tumor. That's really powerful, isn't it? So that means this Ty compound is selecting those cancer stem cell without affecting their original function, and then really powerful the tool to enrich those cells, kind of things. So up to here, everything was good. Now, next step of the figuring out those mechanisms is really challenging. So if it's a drug molecule, so looks like here, this is drug molecule. Most common technique is affinity matrix. Sounds like a fancy technique, but it's just quite rough. Let's just say affinity matrix, usually we make on the surface of the dextran or the cephadex carbohydrate polymer, and then attach those molecules on the surface like this. This looks too close to the bead, but actually it's not. Small molecule, you can think this bead is much bigger than cells. And small molecule should be much smaller than this. That means only very surface of this, the affinity matrix, the bead, can be modified with the molecules. And then we most likely we assume that the proteins inside the cell is like a fish swimming in the sea. This is kind of a picture we have. But to do this the affinity matrix, prepare for the affinity matrix, and you need to break up the cells. And by adding a lot of detergent and collecting those proteins and apply them to this affinity matrix to figure out which fish has good appetite for this bait, right? So that is quite artificial situation, and a lot of fishes maybe they die or denatured. So their binding to this surface may not be specific, not like bound by their the biting. It's just maybe exposed hydrophobic area kind of things. So with this, a lot of the potential problem, we thought we can do a little better trick. Here, our dyes are the fluorescent. That means these fluorescent molecules can go inside the cell, live cells, and then they can act inside there. If I show you the structure of TiY again, here, this part is the fluorescence part. This is the diversity part. If you look at this structure, chemically speaking, this is a chloroacetyl, which is reactive to cysteine motif. That means this is kind of reactive motif at the end. Then maybe this compound can go inside the cell to get those targets without breaking up the cells. That's what is thought. So that means in the biological system, in the cell, if you send this molecule in, it goes in and then only bind to the target protein. And maybe hopefully the target protein is stained and covalently. Then if you break up the cell, still that's okay. Then you can get those the proteins size, and even by analyzing these sequences, you can get the identity of this protein. So if it's very successful, we had several cases of a successful other study. Now you got the binding target. It's a happy ending, but sometimes, or actually many times, we see some funny other situations. Maybe 10% of those dyes bound to some protein like this, but 90% of other dyes are free compound in the cell. That means not only this fish, it's a whole cell that somehow they are stained, and the compounds is simply like just hang around without the binding specifically to any target. So then, could we still claim that this bound protein is really the target protein of this molecule? So we thought we need the different other mechanism. So we started to think about maybe if there is any transporter which can transport this molecule into the cell, and just the molecules are the hang around inside the cell, if they can report their existence, then still they can act as a probes. That's something we started to think. And we thought this kind of the target can be gating target. Now, keeping this in mind, we tried the search that kind of mechanisms. So this is a second example with the activated macrophages. Activated macrophages is the macrophage is a one type of the immune cells, uh, and they have a resting state. And if they are activated, they change into more aggressive the phenotype. So we thought not only this macrophage is a phagocyte. That means they can swallow many things. Maybe easy to find phagocyte, the selective. Even we are not sure whether that is a selective or not. But anyway, phagocyte is swallowing a lot of things. So we thought maybe we may need to activate the macrophage only selective the probe, not the resting state. Then we can get more selective the imaging the probe. It was quite successful, and we found one compound, the CD green 16. So as you see, so we give the compound code with a number similar to antibody or the CD number, and then with the color code together. So this means this compound is green compound, is the 16th compound we made so far. So biggest number we have for now is about the CD20. Now this compound, if you make the atherosclerosis model with the genetic, the knockout, and the high-fat diet, and they inject this molecule through tail vein, then we could demonstrate that this molecule goes in and then stain this vulnerable site of the atherosclerosis, the position. That is good. And then we got some indirect evidence that seems like this molecule does not have a binding target. And we started systematic search for the gating target. To do that, we use the CRISPR systems. CRISPR is originally it is a gene editing tool, right? If it has recognizing sequence with this the single guide RNA, then it goes in into the cell and find that sequence in the DNA and clip. Now, that cleavaging enzyme is a Cas9. If you modify the Cas9 into inactive form, deactivated the Cas9. Now, it can keep the ability to bind, but it cannot cleave. Here, you can add either activating domain or suppressed domain here. Then you can increase the gene expression or suppress the gene expression. In this case, we use the activation system. So this modified version we call CRISPR-A. Using this, we tried to express the transporters. So pumping transporters SLC, we have about 400 SLCs in the body. To increase those chances, we selected 10 promoter sequences. So we made about 4,000 other library and built it into the cells. That means each single cells they express, we assume, express one transporter protein. And if one cell, it that express that specific transporter which can uptake this CD-16, they can be stained. And we select them out and then repeat this enrichment that we hope that out of 400 of the targets, maybe we have a chance to find out that specific transporter which can be responsible for this selectivity. So by doing this kind of a selection and regrowing after 6 rounds, now you can see, so comparing to control cells, majority of cells are positive. And by analyzing these sequences, yes, we could find one transporter, SLC18B1. This protein is responsible. This protein is not expressed in the resting macrophage, slowly induced during the activation, and only activated macrophages express this protein and they pump in the CD-16, and this is the mechanism of selectivity of CD-16 to activate the macrophages. Now this is the interim summary. Uh, Most of those probes compounds, we believe that they have a binding target inside the cell. But we found that there are other mechanisms we can use, pump in or pump out. If it's a pump in, most likely those materials, materials are similar to nutrients. If it's a pump out, maybe they are toxic molecules. That's why for pump in, they don't use the energy. They just use the gradient of the concentration. For this is the SHE transporters. Pump out, they use ATP. This is ABC proteins because this is for toxicity, the clearance. They use energy to pump this out. Original concept, there may be some binding target, maybe holding oriented lifestyle distinction. This is kind of a conventional the concept. So this is what we call hold mechanism. This case, there is a no binding, no holding, but there is gating. So maybe you can call this gating oriented, maybe gold mechanism. So this is the two examples that we found and then established so far. And then let me show you two more examples with still different mechanisms. For the next challenge, we selected T cell and B cell as our target. These two cells, they are very similar in terms of size and shape. It's almost impossible to discriminate without the antibody's help. So for this screening, we collected those cells from spleen and then tried to do this kind of the imaging-based screening again. Now, blue color is the nucleus, and we tried either B cell or T cell. First hit we found was B cells. And here we gave the compound name as a CD green B. Usually we give the number, but in this case, either T or B. So we hope that if we can get the T cell with the green, then we can call CD green T, and maybe red one, CD red bull, kind of thing we wanted to give a name. But so far we have a CD green B. And with this compound, without antibody, you can see this is B cell, this is T cells. We can discriminate them. And then among those mixtures, this is the CD green B treated cells. And T cells, we don't have the compound yet. So this is antibody for T cells. Then you can clearly see those imaging is exclusive to each other. And then utilizing this one, now we tried to get the target ID. Now, if you look at those compound structure here, so with this color, we gave the name as CD green B. Now it looks like it has a long chain here and this reactive group on top. That means we designed this molecule to get the binding target in the beginning, but seems like there is no binding target because even though you remove this, selectivity is still there. And this molecule with this long, the heavy chain, we thought maybe it may bind to the membrane. Yeah, it was true. So with comparing with the other membrane dye, seems like it's a membrane binding. Now we thought there are two possibilities. One is maybe membrane protein is the target, or maybe membrane itself may have some clues for the selectivity. Then we analyzed the lipidomic, the structure, lipidomic analysis we performed for T cells and B cells, and found made two interesting observations. First one is T cell. This is T cell. This is B cell. T cells membrane is a little thicker than B cells by 1 carbon or 2 carbon lengths. Second of all, cholesterol amount is almost double in T cells than in B cells. And we thought maybe B cell is thinner, and then we checked those cholesterol effect. If you add the cholesterol further, here in this case, the different chain lengths, the shorter, similar to B cells, is brighter. Longer one, the weaker. It's a similar observation to what we have seen from the T cell and B cell. And then we added the cholesterol, and by adding cholesterol, is the further and further decreased. And by analyzing the different properties here, before that, we tried to make the mimicking the membrane of B cells and T cells without any proteins. So made giant the vesicle, and then fill that with the blue dye, and this one is the B cell model, and then another vesicle filled with the red color, and mix them together, and then here we added CD green B. Then it shows, even though they exist together, this CD green B bind only to B cell model, not to T cells. This model is made by, of course, by the different chain lengths and also different cholesterol contents. So this is B cell is around here, T cell is around here. So this means that even without any protein compositions, only the lipid composition are the control, we can differentiate two types of cells. So this is the evidence that maybe by utilizing this kind of a concept, this compound originally fluorescent in the water is aggregated. And then if you meet with a membrane structure on the cell surface, they start to integrate into them. And this integration is kinetically different between softer, more flexible B cell versus more rigid, more thicker T cells. They compete to each other, and B cell has a better chance to take up those compounds. So this is a totally new mechanism, unheard before. So this is kind of a lipid-oriented lifestyle distinction, maybe you can call without. So this is third example. Now I'm moving into the last example. So by looking at these immune systems, in the beginning we started with the T cell and B cell, the discrimination, and this macrophage for the activated and non-activated one. Now we thought we wanted to look at this granulocyte. Granulocyte, there are neutrophil, basophil, and eosinophil. Basophil means it likes space. Eosinophil means it likes acid, right? And even this name is given by the staining pattern by acid dye and base dye. It's very funny. It's similar to the gram-positive, gram-negative staining. Even before we knew about those biological reasons, why they show those difference, we used the chemical which is stained selectively, and we classified those cells. It happens to our immune cells too. So I thought we can discriminate these three. Current, this H&E are the staining patterns. They need to fix the cells again. So we wanted to keep the cells alive and still wanted to differentiate. So we found one compound which can stain neutrophil selectively. That's the example I want to show you. With this platform, now to obtain enough numbers of cells, we selected human blood. Mouse blood, you can take maximum from one mouse, less than 1 ml. Spleen can provide you the T cell and B cells, but this granulocyte is very difficult to get. So we use the human cells, means myself and my people, only volunteers. And in this the blood cells, at least we have those cells, lymphocytes here, T cell and B cell, and monocytes and granulocytes. This is separated by scattering. Okay. Here by adding, use the whole blood as a screening setup. Here we added compound, and then same scattering side scattering on this axis, and this axis is a fluorescence axis. Here now you can see The lymphocyte moved a little up, but not very far. Monocyte is similar, and among those granulocyte, some population shifted much further, and the other population still stays. I didn't tell you here, but neutrophil is occupying almost 80-90% of the whole populations, and the basophil and eosinophil is around a few other percent. So we suspect this may be the neutrophil here, and this may be either the eosinophil or basophil. Again, here if you look at these compounds is also shifted, but the degree of this shifting is neutrophil is almost 10 times stronger. And by taking these populations, we analyzed this here and here with the antibody, and we confirmed that this is neutrophil antibody. These positive cells are all neutrophils. That means these minor populations, these are eosinophil plus basophil stays here. So we got neutrophil compound, and then by analyzing those structure, as you can see here, we tried to make this small library. This library has a different chain length here and here, and best compounds is around here. And then by analyzing those the stain index, we selected the best one, and then gave the compound the name as a neutral the G. Now, by looking at this structure, now you can guess this molecule is very similar to fatty acid, right? So in the beginning, we targeted this molecule, we designed this molecule as a gold probe. Maybe some cells may prefer to take more fatty acid than the other cells, something like that. Now, this is whole blood. If you add this compound in here, before that, you stain nucleus, and there are many cells, but only 3 cells are have nucleus. And here you add the neutrophil G, only 1 cells are stained, and then the neutrophil antibody to confirm. So, as an end result, out of many cells, this only single cell, this is a neutrophil. The others are not. Maybe these are the red blood cells, and by shape, this may be the monocyte. And there's a lot of cells here, but this is the image we could show out of a mixture of many other cells. This is a neutrophil. Now, this staining pattern looks like some lipid droplet, isn't it? So we checked whether that's really lipid droplet or not, and neutrophil with the Nile red, so they are overlapped together. Okay, this is a lipid droplet staining, and we isolated the eosinophil and stained to check whether they have a lipid droplet or not. They do. They have a lipid droplet, but they are not stained by neutrophil. Situation was the same for the basophil. They make a lipid droplet too, but they are not stained. So we thought maybe the biochemical synthesis pathway to make lipid droplet may be different, and we checked those expression level of the key enzymes in these metabolic pathways. And neutrophil has high expression of ACSL1, DGAT2 comparing to other cells, right? And we tried to make a model for the biochemical pathways. So maybe first player can be maybe CD36, which can transporter for this fatty acid. And then ACSL, so by adding those coenzyme A here, and then DGAT is converting this into final triacylglycerol. So this is kind of a pathways, and then we tried to confirm whether this is the case or not. Now, one problem here is we need control. The control is either the neutrophil or, the neutrophil is a positive cell, then eosinophil or basophil should be the control. The amount of those cells are too small. To collect them, you need to use max expensive the mixture of antibodies, and we need to provide a lot of blood. So that was a little too difficult. So we thought maybe a little smart way. So we decided to use the whole cell screening the format as the validation format too. So at least these cells, basophil and the eosinophil, they are shifted too. And then this is a little further shifted. And here we tried to add inhibitors for this. Then see how this the staining pattern is changed. So here, this is a bright population of a neutrophil, and this is a mixture of eosinophil and basophil. Now we added those inhibitors for these three players, and you can see here, blue one is a neutrophil. By adding neutrophil pattern is reduced. DJT, same thing. And eosinophil and basophil is not changing. That means these enzymes are only important in neutrophil, not here. CD36, not much changes. So using this data, we could make the conclusion that CD36 is shared by all cell types. But on these pathways, ACSL and DGAT pathways, for some reason, this pathway is only activated, the more activated than other cells, only in neutrophils. And eosinophil and basophil may utilize different pathways through this and then go into this the lipid droplet synthesis, but not sharing this. That means we could hijack the metabolism pathways only selectively overexpressed in neutrophil and distinguish those cell types. So that's why we call this approach as metabolism-oriented live cell distinctions. So as a summary for this approach, neutrophil we found for the first time neutrophil G the probe. Basophil previously we had some indirect way by developing histamine the sensor. So histamine is the secretion molecule by basophil. We checked this molecule and seems like it can selectively stain basophil. Eosinophil we couldn't find yet, so we just used the antibody and then mix these three cells together and stain them. So even though the staining pattern is a little different, but by collecting these cells together and looks it's not really big deal, but these cells are still alive. And this is the first demonstration that alive three cell types could be recognized in one set of the probe cells. And you may ask, how do you know whether they are really alive? So here is one movie. So this is a neutrophil stained by the membrane dye and the neutrophil G. This is nucleus stained the bacteria. Then if you look at here, that means neutrophil is still healthy and functional. And you may have seen these similar pictures like a bright field images. But in that case, you cannot say the identity of those cells. It can be either basophil or eosinophil or maybe macrophages, right? So this is the first example of a fluorescence version of the live cell imaging of a functional the neutrophils. So this is my summary of the slide. So by developing chemical tools, we wanted to develop the cell selective probe, if possible, using different mechanisms. We started with the holding mechanism or binding mechanism. And then we expanded those strategies into gold mechanism, which is also occupying the big class of those mechanisms. Relatively the rare, but there is a possibility of utilizing lipid structure or membrane structure. And metabolism is another big branch, we feel. So utilizing this kind of mechanism, of course, we are trying to expand this further to different mechanisms like viscosity, polarity, mitochondrial membrane potential, and temperature difference kind of things we are trying. So far we have worked on the tumor microenvironment as a main target. Now slowly I'm shifting my interest into aging. So comparing to the tumor microenvironment, which has maybe more than 10 different other players, aging so far I feel it's simpler. You have a young cell and older cell. All you need is to distinguish them. And then using that probes, we have a chance to find out the new biomarkers. So far, maybe comparing to the tumor research, aging research is quite booming up, but still those available biomarkers are not the many yet. So if possible, using those biomarkers, if they are the biomarker is responsible for causing aging, maybe by attacking those targets, hopefully we can reverse it. So this is the dream and picture for the next 10 years of our research. Yeah, so this is my collaborators. As a chemist, our role is providing the chemical tools, And maybe we do a little biology, but actually we are not hardcore biologists. So we need a lot of help for providing problems and prove the validity or value of our probes. And always we are looking for new collaborators. So this is the end slide of my talk, and thank you very much for your attention and welcome any questions. Thank you.